Welcome back to Monkeys on Typewriters on YouTube. We're continuing with our series entitled How Does This End? And this is technically part three, although you may recall that part two is divided into a few different sub episodes. Links will all be down in the description. So this time we're actually moving on to talking about SARS-CoV-2, and hopefully a lot of the background concepts, the historical context, and all the rest of that will be apparent as we move forward talking about the current pandemic. I'm not going to promise that it's all going to be good news here, but it will give us some better understanding of where we think things might be heading with the current pandemic virus. So, Arjit, I'll hand it over to you, and looking forward to hearing about SARS-CoV-2, and also not looking forward to it in many ways.
Thanks for the cheerful intro, Ryan. Relatively cheerful compared to what's coming next. So we're going to talk about what happens with SARS-CoV-2, and it's a trope, I think, that you often hear when people talk about pandemics, that no one can really predict what's going to happen with evolution. Who knows what will happen? Maybe we'll get lucky. As Trump once said, one day you'll see it'll just disappear. Well, in fact, it has disappeared, to be honest. I mean, they've disappeared it. Yesterday we had someone resigning from the CDC's panel that reports hospitalizations in protest. So it's been disappeared. But unlike the commissar, when Stalin disappeared the commissar stayed buried. Infectious diseases don't do that. So you can bury an infectious disease all you want. You can move it off the front page, but it'll do what it needs to do. What we saw in the earlier episodes was two major themes. And one is evolution has certain predictable themes. You see the same things over and over again when you look at biology. And when you look at historical pandemics, you see certain themes as well. So when you look at it from an evolutionary standpoint or a biological standpoint, we've seen that host and parasite interactions for pandemic pathogens follows what's called the Red Queen's race. So you get this sort of jumping around of contagiousness. You get sort of multiple rounds of adaptation on both sides. So it's very much an adversarial battle. And then on the other hand, what we saw from history was that those societies that made the choice to live with pathogens or lived with pathogens because they had no other choice were extremely vulnerable to flare-ups. We saw people made the choice to live with rats, and their reward for learning to live with the bubonic plague was repeated waves of massive plague-induced mortality. We saw that when smallpox swept through, smallpox, which was an endemic disease in the old world, swept through the new world, it very much behaved as a pandemic would, causing societal collapse. So today we're going to now sort of apply those kind of concepts to SARS-CoV-2 and with the lens of both history and biology, ask some fundamental questions about SARS-CoV-2. And we've been hinting at this through the entire series, but now we're really going to dig into it. So the first thing we should talk about is this trope of evolution is not predictable. So many of you who follow me on Twitter know this, but a bunch of collaborators and I we had an ad hoc ragtag band of collaborators starting in the summer of 2020. We have published about 24 papers on SARS-CoV-2. And there's a video pinned on my profile from October of 2020. If you're ever in the mood, get yourself a stiff drink or a tub of ice cream, whichever poison you prefer, and watch that video because it's really depressing. We basically went through in October of 2020 and we sort of talked about what our research was showing about what the future would hold. And unfortunately, it aged very well. So let's talk about those predictions and let's talk about how predictable the evolutionary trajectory of SARS-CoV-2 has been so far, because this is a really important point. Yes, the exact mutations, a lot of people put effort into understanding the exact mutations and what mutations are coming and going. That's one way to look at it. And when you look at it at that level of granularity, evolution is highly unpredictable. But there are certain larger themes. And if you use the right lens, you can sort of make some very clear-cut predictions very far in advance of the actual events. And one such set of predictions was what we made back in 2020. So in the summer of 2020, we predicted that the virus would evolve rapidly to evade antibodies. And so this paper on the top right which we posted in on medRxiv in fall of 2020, late fall of 2020, was the product of work we were doing in the summer of 2020. I can tell you that the summer of 2020, especially when everyone was like, well, this will be over soon, was a depressing and disorienting time for us as a team. And this was the aha moment or the oh shit moment, if you will. We compared the spike proteins mutations. One of the folks on our team mapped the binding context for the ACE2 receptor binding and the receptor binding domain of SARS-CoV-2. So this is the RBD over here. And in green are the contacts on the SARS-CoV-2 spike protein receptor binding domain that actually contact with ACE2. So if you look at the same RBD on the right-hand side, and now look at the antigenic determinants for natural immune system response, this is what you see. So what you see is that the naturally occurring neutralizing antibodies cluster in a narrow region of the spike protein that overlaps with the binding contacts for ACE2. This is not surprising. This is what you expect. What's happening is the neutralizing antibodies are sitting on that receptor binding domain and occluding it sterically, blocking it sterically from binding. So literally sort of once it's sitting there, nobody else can sit in its seat, so to speak. Now, we also looked at the impact of mutations on this. We said, okay, well, if we now look at mutations for binding affinity of ACE2, what happens? And a number of labs at that time and since then have done some phenomenal work. A lot of this came from the Bloom lab. Some of this came from Regeneron, which is a pharma company. And they published these papers as preprints in the summer of 2020. So we used the preprint data at the time, subsequently published, and we said, all right, well, what happens with the mutations? So we plotted those back onto the RBD surface. And what we noticed was that the binding contacts, the darker the yellow, which is kind of a weird thing to say, but the darker the yellow, the more strongly those binding contacts, those contacts impact binding. So what you can see is these very quote unquote dark yellow amino acids over here with the red arrows. These amino acids are the ones that are responsible for the binding. So on the left-hand side, you see this green surface for the binding, overall binding surface for ACE2 and the receptor binding domain. On the right-hand side, you see which binding contacts have been impacted or which mutations have the most impact on the binding affinity. So only a small number of residues within the receptor binding domain are really crucial for binding to ACE2. Okay, so that's not great, right? Because that means that what would the consequence be if you look at those receptor binding mutations? What would the consequence be on antibody recognition? That begs the question, right? So when you look at the antigenic determinants of natural immune response and you overlap it with the impact of mutations on binding affinity, what you notice is individual mutations within the region of the spike that is preferentially targeted by neutralizing antibodies don't abolish binding to ACE2. This was our oh shit moment. When we saw this pair of images in the summer of 2020, we realized as a team that this was not going to go how they were saying it was going to go. Because right here, what you notice is that the virus's ability to evade the natural immune response while still binding to ACE2 is really good. As you can see, there's a sea of dark yellow and lighter yellow, which is all amino acids that would make a positive contribution to ACE2 binding that are not impacted by the antigenic determinants of the natural immune response. In other words, the virus, we realized, was free to mutate key residues that would be responsible for the immune response while not negatively impacting RBD binding. This, you remember, was at a time when Fauci was out there saying, we don't think that this virus will evolve quickly. We had all these experts platformed on TV and in the news. When the first mutants came out in the summer, in August of 2020, there were the first mutants that showed up. D614G was the first mutation. And Betty Kober's group was like, oh shit, there's D614G, look at this. It's a novel mutant that's shown up in a matter of months. And everybody kind of jumped all over that and was like, well, this is too preliminary. We don't know what this means. We don't understand the significance. Let's not panic, right? So the calm mongering happened in full force. But at that time, it was kind of obvious from this graph. And this data was all in the public domain. The experts who had spent their time on TV and talking to the press may have spent their time better, one may argue, just doing this simple exercise of comparing the binding residues. Even worse, when we looked at the experimentally determined mutations that were in preprints at the time, we noticed that the experimentally determined mutations that were called escape mutations, and I think the Bloom Lab or Regeneron, one of these groups did this, the escape mutations did not impact ACE2 binding. Now, what's funny about the papers that these things were reported in, they didn't do side-by-side comparisons, or maybe they did by themselves. You know, the labs may have done this, but they didn't show it in the papers. So they were all about, oh, look, here are these experimentally determined mutations, so we can target out this stuff to make escape-proof antibodies, which is all well and dandy. But actually, there's a bigger question here, and we'll get to that in a minute. If these experimentally determined escape mutations existed, not impacting binding ACE2, the next question was what happens to these mutations in the wild? So we looked at GISAID, which is the database of mutations, and we said, well, what variants are showing up in GISAID? This is the summer of 2020, and we're saying what variants are showing up in GISAID. In the summer of 2020, what we noticed is there was a wide range of tolerated variants in GISAID that essentially were going to impact antibody binding, but not impact RBD binding. Now, as you can see, even in the summer of 2020, this little cluster over here, which is critical for binding, was not showing up in GISAID. Later on, this cluster showed up as well. So, but even early on, you could see that there was plenty of stuff in the sort of medium colored red, not the deepest red, but the medium colored red, where you had opportunities for the virus to evade neutralizing antibodies, while on the right, being tolerated, being viable. So we reasoned that the evolutionary cost of resistance was very low for the virus, because the evolutionary cost of resistance to neutralizing antibodies involved mutating residues that then made the virus's RBD not be recognized by neutralizing antibodies, while at the same time allowing the virus to bind the RBD, the ACE2 protein. And so what we noticed was that in the summer of 2020, there were already variants kicking around in GISAID, meaning in the human population at that time, that were theoretically capable of doing this. So then we asked a simple question. We used an evolutionary model, and we said, all right, well, looking at sort of the frequency of single or double mutants or triple mutants, how long would it take for essentially for a population that was, for example, treated with a neutralizing antibody to lose effectiveness of that neutralizing antibody? So what we noticed was that mutations that were, so what you see over here in the different lines is the number of infected individuals. We varied that from 5 million to 640 million. So 640 million individuals infected at any given time. At the time, we were sort of criticized for using that 640 million number, because people were like, well, you're never going to have 640 million people infected at the same time. Unfortunately, that's roughly where we are right now. What we noticed was that depending on the number of people infected and depending on the number of mutations, the virus would evolve resistance to neutralizing antibodies very, very quickly. So I'll explain this graph on the top left first. So this is the log 10 of the number of months till the mutant is established in the population. If you had a mutant that through a single amino acid change was capable of being resistant to a neutralizing antibody, that single mutant would show up in the population by about, this is negative 1.5 months. So those mutants already existed. So first off, if there were any neutralizing antibodies that could be defeated by a single mutation, those were toast already. Then we said, okay, what about neutralizing antibodies that take 2 mutations to defeat? And we found, well, some of those will take about 1 month. Some of those could take 10 months. So double mutants were looking pretty iffy as well. So in other words, antibodies that required 2 mutations to escape were looking pretty iffy as well. Antibodies that required 3 mutations to escape were going to take a bit longer, and antibodies that would require 4 mutations to escape were going to could take a long time. Now, we got the math a little bit wrong simply because we didn't realize how much there were other factors that we weren't considering, such as recombination or other things. But the premise was that from standing genetic variation alone, which is the only thing we were looking at, how long would it take the virus to defeat neutralizing antibodies out there? And we were looking in this case at the neutralizing antibodies that were then due to come on the market. So what we said is the neutralizing antibodies that are due to come on the market will get defeated in a matter of months. Indeed, they did. Not only did they get defeated in a matter of months and withdrawn, as we all know, but the evolution-proof vaccine neutralizing antibodies, monoclonals that were made and sold, evolution-proof like sotrovimab, also took a matter of months on the market to get defeated. There's a whole story about that, but this whole idea of things being evolution-proof is based on sort of the wrong way of thinking about the problem. What they do is they look at the sort of the ancient history of the virus and say, well, if this particular residue in the RBD of the spike protein of the virus was not changed as the virus jumped from bats to humans, then that's the one we should target. That only works if what you're trying to block is a transition from bats to humans. It doesn't work if what you're trying to block is a transition from transmission from one human being to the next. So because they were using the wrong logic for evolution-proof vaccine or evolution-proof monoclonals, those so-called evolution-proof monoclonals lasted a matter of months, as we predicted. Again, this was in the summer and fall of 2020. So this is really creepy, right? But working from this assumption, then we said, okay, well, what is the bigger picture here? So we then asked the question, what would happen if you modeled out using an SEIR model, which is a standard epidemiological model? What would happen in terms of the population as a whole with vaccine rollout? And we were especially interested in vaccines that had a shorter half-life than lasting forever. So at the time, if you remember, there were famous experts such as Francois Bayou and others. There's a whole T-cell mafia that were predicting that because of T-cells or because of this or that or the other magic, immunity to COVID would last a lifetime and vaccination would not be needed more than once in your lifetime. These were all things that were getting said and they were getting a tremendous amount of play in the news media. We said, okay, well, based on what we know, that's not true. So let's ask the question, what will happen? So we said, okay, well, if you roll out a vaccine that essentially, a certain percentage of the population takes between 50 and 100%, and the vaccine is highly, highly efficient against mortality, up to 100%. We varied this between 80 and 100%. And then you look at an SEIR model where people are getting infected repeatedly on an 18-month interval with an R naught of 5.7. So this was a simulation that we were doing, and the conditions we were using, unfortunately, are very, very close to what happened in the real world, except that we don't have this kind of compliance anymore, and the R naught is probably a good deal higher than 5.7. But what we noticed an interesting thing, that even with this kind of a highly effective vaccine, with a lot of the population taking it, we would expect to see a huge number of deaths in the first year following rollout. And that, in fact, was what was seen. In January of 2021 in the US, we had 382,000 deaths. The US had about 50% compliance with the vaccine in the first year. And by the end of 2021, we had, give or take, about 1 million deaths. So that was 600,000 deaths, almost on the nose, unfortunately. But not only that, what we were able to show was that this kind of a vaccination strategy was not going to work at all. We showed in other papers that essentially if you vaccinate, if you vaccine relax, there are other consequences. But remember, this is all in the context of a moment in time. This all looks very obvious now, but it wasn't obvious then, right? This was January of 2021 when we put this out as a preprint. And at somewhere in that preprint, we used the term endless waves of disease. These were the headlines at that time in, the Washington Post and New York Times and so on, right? It was like the cavalry is coming. There was literally, I forget, I think Wall Street Journal was like the vaccine cavalry will be here, or the T-cell cavalry is here. And there was a lot of talk about how immunity was going to be sort of lifelong, and then vaccination was going to end the pandemic, right? Oh yeah, here we go. But the medical cavalry is coming. Yeah. So, I may be biased being an Indian, but this was much more like Custer's last stand than it was like, than it was the cavalry of the Old West. So when it came down to it, what we saw in this situation was that we were going to end up with a long-term um, pandemic scenario. We also predicted that non-compliance would be a huge issue. So here we did some modeling using game theory, and what we said was, okay, if we reason that people will uh, comply or not with COVID mitigation measures of any sort, um, based on whether or not it benefits them, right? If they're rational about it, then they look at the cost of the intervention relative to the cost of infection, and depending on the efficacy of intervention, we ask the question, in what zones is compliance favored? In what zones is non-compliance favored? And so essentially the question we were asking is, if you're asking people to comply with COVID disease mitigation measures, how likely are they to comply with it? And so the red part is the part where it makes sense to not comply, and the blue part is the part where it makes sense from a game theory perspective for people optimizing selfishly to comply. Right? And what we noticed was that the entire square was basically red. That the minute you went down the altruism route and you said, let's let's follow, let's wear masks or let's vaccinate or whatever, let's socially distance to protect the vulnerable, people will throw the vulnerable right under the bus. Again, this was in December of 2020. And so the paper title was Individually Optimal Choices Can Be Collectively Disastrous in COVID-19 Disease Control. And the point we were making here was that if you have a public health messaging sort of framework that is centered on altruism, this will backfire. As we know, public health messaging was centered on altruism. We were lied to about the cost of COVID to us individually. So the perception of actually the cost of intervention relative to cost of infection, we're actually right down here somewhere, because it's much, much worse to get COVID than it is to be vaccinated, for example. But people think that they're here somewhere because we've been told that COVID is not a big deal unless you're vulnerable, unless you're sick. So now when people mask, they are self-identifying as being vulnerable. They are self-identifying as being one of the people for whom the lockdown was done. They are self-identifying as being part of the quote unquote problem of COVID, right? So in fact, what's ended up happening is through this othering of other people as being vulnerable, through this message that COVID is other people's problem, public health has been very successful at getting people to ignore COVID. And unfortunately, I always think that we write these papers so that policymakers can read them and do something useful with them. It's always possible that the policymakers read this and took the wrong message away, because the degree of effectiveness of altruism in pushing people as a message, in pushing people to go and ignore COVID to the detriment of their own health, was laid out fairly clearly here. On the right-hand side, you can see that essentially you need a very, very high percentage of compliant population in order to get a vaccine to work. However, if the vaccine had been more effective, we would have had a much lower requirement for that. So partially effective vaccines were very much part of the problem. And even to this date, there's been no attempt made to improve the effectiveness of the vaccines. There's been no attempt made to improve the effectiveness of the public health messaging. Because the idea has been, well, just let it rip, right? And the idea somehow is that letting it rip and learning to live with the disease will bring with it some reward. So the first time we saw letting it rip, the vaccine relax mentality, was in the summer of 21. In the summer of 21, in May 14, 2021, we posted on medRxiv a paper that showed that if you vaccine relax, you will get a vaccine-driven wave. It was kind of that simple. And the only thing is we guessed the wave wrong. We thought it was going to be, I believe, beta, the one that was in Brazil. And it wasn't, it was Delta, which was still sort of simmering in India at the time that we wrote the paper. But so essentially what we did is we had two populations. One is the infected and recovered from the wild type, and the other is a population that was infected and or recovered from a variant wave. And what we showed is even small numbers of variant cases, if you drop mitigations in a partially vaccinated population, would lead to an explosion. And that indeed is what we saw, right? So we posted this thing in May 2021, and through the summer of 2021, we saw an explosion of Delta cases, and that was your first variant-driven wave. So what this came down to, again, we're talking about how predictable this virus has been. The virus has been pretty damn predictable, and the consequences have been pretty damn predictable. And we really sort of, we really experienced a lot of personal frustration, the team, all of us in the team, when we saw this shit in the summer of 2021, where, Rochelle Walensky was like, oh, the virus threw us a curveball. Like anything is a curveball if you're sufficiently ignorant or unprepared. And I think that was, Walensky's motto was, oh, it's a curveball. And, every other, as with every other CDC director, she was quietly pushing for pro-infection policies while, wrapping it in the mantle of altruism. And it very much was not a pandemic of the unvaccinated. It was very much a case because there were so many individuals who had been infected. Um, there was going to be an increased number of cases with reinfection. We showed in the summer of 2020 that that was the case, and indeed it was. So yeah, it's not that the science changed as the virus changed. It's that the science said that this was what would happen all along. And at the end of the day, evolution, even if it's not predictable in the minutiae of it or in the molecular details of it, evolution works in predictable ways. Um, and in fact, even to the point where you can predict what will happen if you let something evolve. So again, this was in the spring of 2021 when we posted this on medRxiv, and then we published it later that year. What we showed that was in this particular paper was that with long-term infections, you can expect over time you would get the emergence of resistance. And we showed essentially long-term infections would be a very efficient engine of driving resistance. So what we looked at over here was we looked at sort of the selection coefficient within individuals, and we said, okay, well, if you have a selection coefficient within individuals that selects for resistance to neutralizing antibodies, how long would it take for those variants to be out in the wild? And what we noticed was that essentially, as soon as you cross like 5, 7, 10 days on the right-hand side, even modestly selected for mutants are going to show up at some frequency in the exhalations of a person who is still infectious. So what we notice is that if people are capable of passing on long-term infections, they're going to be basically doing so efficiently. This was posted on medRxiv right around the time as the first reports of intrahost evolution in people was being presented, and there was a theory at the time that said, okay, well, the beta variant and the variant out of South Africa at the time, I forget which one that was. Omicron. This was before Omicron, so this was in the summer of 2021, and by the time Omicron came around, it was fairly clear that this was the mechanism. So the bottom line here is that again, the emergence of Omicron was another one of those curveballs. But if you saw this paper in the summer of 2021, it was fairly clear cut that that would be the mechanism by which the big jumps would happen. So everything we did was done in service of learning to live with the virus, right? And the idea is somehow that if you have a virus kind of like hanging out, in the population, it's going to be fine. Things are going to be fine. And as this creepy AI generated picture suggests, that's not always how it's going to go, right? What you end up in, the situation you end up with is very much a situation where you are at the mercy of what the virus decides to do next. And so we've seen that, right? We have several variant-driven waves of infection a year at unpredictable intervals. So this is just from 21 and 22, right? These gaps are unpredictable. And these are all the result of new variants showing up. So whenever the variant shows up is when everyone gets infected, more or less, right? Or when a chunk of the population gets infected. Modeling from us done in 2021 suggests that the average person will be infected once or twice a year if they take no precautions. Others have done wastewater-based estimation where they found similar numbers. Essentially, on the right-hand side, this is what happens if you're unvaccinated. Depending on how much you reduce your contact, you could end up with as many as 1.2, 1.5 infections a year. Sorry, on the left-hand side. So as many as 2 infections a year. And if you take some precautions, that number decreases. But you really have to drive your contact rate down a lot to have a low risk of being infected every year. That's even true for vaccinated people, as you can see on the right-hand side. So basically, vaccinating and relaxing, and this is assuming vaccination once a year. More or less, if you compare the left and right graphs, what you can see is, roughly speaking, getting vaccinated once a year buys you 1 less infection a year. So getting vaccinated multiple times a year, and we've shown this in a paper, is very likely to reduce your risk of infection massively. I'd say the people I know in my family and among my friends who get vaccinated multiple times a year, very few people have had COVID, and if they have had it, they've had it once. You can and do get unlucky, that's true, but you're going to end up getting it far less frequently if you get vaccinated frequently. But the point is that for most people, the penetration of the vaccine in the population right now is like 10% or 20%. For most people, they're not getting vaccinated anymore. And, you know, public health, not only in the US, but worldwide, is doing everything they can to rely on repeated infections as a way of protecting people against COVID. Well, that's a really bad idea. And here's why. When you get a COVID infection, it starts out in the throat, right? But over time, these infections can be systemic. They initially make their way into the lungs, and very early on, they make their way into the brain. From there, they can make their way into the intestines as well. And then the sequence of events is that at some point, if you have a COVID infection with high enough viral load, it's going to end up in your blood. When it ends up in your blood, it can end up in any organ. Now, this is true for people who are extremely sick, but it also happens with asymptomatic infections. It's been shown in autopsies of people who have died of other causes. When they look at autopsies of people who died with other causes and they test for COVID, they find COVID everywhere. So when they look at organs from people who died of natural causes or died of an accident, and then they look to see what happens with the virus, they find the virus everywhere. So this is really sort of something worth thinking about, because then the question is, what does the virus do when it's in the stomach or when it's in the heart? And the answer is not good. You're not going to like the answer when you hear it. So it actually damages each of these organs as it goes through. And this is sort of a key point that many people have made. There's hundreds of thousands of papers on this topic. Ziad Al-Ali's work shows in long-term studies with electronic health records that there's all kinds of long-term effects from COVID. And very often you'll see these things being sort of described as well. People have this symptom for up to 3 years. No, it's not for up to 3 years. It's for at least 3 years. If you look at the papers, 3 years is where the cutoff is for their for the monitoring. So whenever you read these papers about, this symptom or that symptom persists for up to X months or X years, that always read that as for at least, because that's what the paper is saying. It never shows resolution. And it's not that people don't see resolution of certain symptoms, they do, but it's entirely possible that you end up with persistent infection. So about, I believe, 15% or so of infections are persistent. And so if you get infected with COVID once a year, once every other year, you have a 1 chance in 6 or 1 chance in 7 of having persistent infection. So you do the math for that and figure out how many infections it takes before you are literally learning to live with COVID in your bone marrow, right? So there's a wide range of documented sequelae of COVID. Now, those have been shown in electronic health records, and electronic health records have their weaknesses. But they've also been shown with microscopy. They've been shown in autopsy studies. They've been shown, people have done functional studies where they show, for example, that COVID infects and kills cells of the immune system. COVID infects and kills cells in the kidney or in the GI. So these things have been shown. SARS-CoV-2 as a virus has been directly shown to cause virally mediated pathologies in these organs that it's also been shown to infect. So these are not benign infections. Now, the worst part about this is that sometimes people have seemingly mild acute courses of disease and then have cryptic infection. This has also been demonstrated. And so your cryptic infection can lead to long-term health consequences, even if the acute course of the disease was mild. When people talk about long COVID, they're like, oh, well, look at the percentage of long COVID. It's not increasing. That's true. It's not increasing. But it's still 13.7 million adults in the US as of 2023. And when you look at these questions, what you notice is that long COVID is defined by the CDC extremely narrowly. This is the question that they use to define long COVID, which is tiredness, fatigue, difficulty thinking, forgetfulness, joint or muscle pain, heart palpitations, chest pain, dizziness, menstrual changes, changes to taste or smell, or inability to exercise. That's a really small list compared to this. Notice all the stuff on this that isn't mentioned in that questionnaire. You know why it's not mentioned in the questionnaire? Because people can't test their kidneys themselves. You're not going to know if you have GI tract damage or if you have kidney function loss or liver function loss. That's not something that can be teased out in a questionnaire. So long COVID as such is limited to the symptoms that people perceive readily. And even with that, you still have 13.7 million adults. So if you look at long COVID as a proxy for how many people have persistent infections, then what you see is the proportion of people with persistent infections is not decreasing, at a minimum, not decreasing. There's a study from StatCan that shows that the health burden keeps increasing, has been increasing. If you look at simple questions like remembering, concentrating, or making decisions in the working population, what you see is from the start of the pandemic, that number has been increasing over time. The Canadian studies suggest, bottom right over here, suggest that the number of infections you have is sort of directly proportional to your risk of long COVID. And what they showed was that people with 3 or more infections, and this was in 2023, had about a 30 or 40% chance of having experienced long COVID. Again, long COVID is just the tip of the iceberg here. If you look at the top left over here, that's a figure showing workers who have not been able to work due to sickness. And that trend, and this is British data, that trend essentially has been straight up in Britain for the last, from the beginning of the pandemic. On the right-hand side, you're looking at an illness analysis, just looking at mentions of illness on the AFL website in Australia. And what you see is essentially the number of people looking for this on a Google search has been skyrocketing over the years. In Spain, on the bottom left, the number of people who have chronic health issues has gone up steadily. If you look, it has gone up across all age groups. So if you look at the difference between 2018, 2019 to 2023, you see a gap there of about 20%. So again, the point that somehow all of this is going to be just fine, is very debatable, right? People are getting infected on a regular basis, and that those infections have long-term health consequences. There is no other disease that I can think of that has this many long-term health consequences, where public health says, well, that's pretty chill, like don't worry about it, just get it and you'll be fine. It'll protect you from anything bad. Well, it's a very broad definition of protection at that point, right? It's a very skewed definition when you look at what happens. In a separate talk at a later point, I'll walk through sort of it's not mild, going through what the exact evidence is for at a molecular level. And it's really chilling. When COVID infects neuronal cells, it kills them. You could say, well, if that was the case, would we'd be seeing cognitive problems? Well, people have done cognitive tests on people who've had COVID, and yes, there are cognitive problems. You could say, well, in that case, we'd be seeing it in the population. And yes, indeed, IQ tests, scores are falling on IQ tests, scores are falling on tests that are associated with cognitive ability, such as the SATs. So you are seeing population-wide changes. The impact of these things is being felt at every step along the way, whether you look at cardiac events or whether you look at testing scores, you see population level impacts already. Remember that by the time something impacts the population as a whole, you have a huge impact already in order to be able to see the signal above the noise of what happens in a population in general. It's not like there's a flat baseline, for example, of for these things. So if you look over here, just as one example, if you look at sickness, right? The sickness baseline is not flat from 1919 to 2019. If you weren't looking at the right part of the graph, you'd say, well, it's very noisy, right? And indeed it is noisy. What you end up seeing when you have a really strong signal can exceed that, but it doesn't have to exceed that to this extent for it to be a problem. You could end up seeing a 50% increase or 100% increase, and that would already represent a huge surplus. So these things, the logic that somehow these things are negligible is beyond baffling. Countries have stopped reporting COVID deaths at this point. But if you look at the excess death numbers, and this is through the fall of 2024, you can see that the excess death estimates are still climbing worldwide. So these are the deaths over and above the deaths that regularly happen. And mysteriously, the number of excess deaths have continued to climb on a year-over-year basis. Now, again, COVID infects a very wide range of organ systems. 1 out of 5 infections is persistent, or 1 out of 7 infections is persistent, and you're going to keep getting COVID your whole life. It's not implausible to say that at some point people's luck runs out and they end up with a COVID infection that is progressive or is debilitating. So the idea that somehow, again, that COVID infections, if they are mild in the acute course, have no long-term consequences is something that has been promulgated by public health. And that promulgation, as we've talked about, has led to sort of a false sense of security. And that false sense of security has led people to throw the vulnerable under the bus. But the key point is this: if you've taken anything away from what we've been talking about in the last 20 minutes, it's that we're all vulnerable. Everyone is vulnerable to this disease, and repeatedly so. And as you keep getting infected, the chances are that your chances of a bad outcome just keep increasing, not only probabilistically, but you'll also start losing different aspects of your health, which will compound. So, for example, COVID increases the risk of type 2 diabetes. If you get type 2 diabetes, that's not great. But what makes it even worse is that COVID outcomes for people with type 2 diabetes are worse than without. Now you're in the vulnerable pool with that COVID-induced type 2 diabetes. So COVID has made you vulnerable. So the idea somehow that, there's a vulnerable subset of the population has been tremendously damaging to us societally. It's shown us exactly sort of how much societal solidarity there is, but also it's been damaging to us as individuals because we believe we're invulnerable to this. And if you look at it, the number of children dying from COVID or sickened by COVID is extraordinarily high compared to other infectious diseases. And we're chill about this. So where does this leave us? Like what happens next from an evolutionary standpoint? It's bad enough the way things are, but are there other consequences from an evolutionary standpoint? Well, again, we're talking hundreds of millions of infections at any given time. So you have an enormous viral quasi-species. So just very roughly, on a daily basis, 30 million people are infected. So if you do a little bit of quick math, you get 2 quintillion viral particles at any given moment in time. So I believe that fits inside a Coke can. But nonetheless, it's 10 times more than the number of grains of sand on the planet. So it's a very, very large population of viral particles. Now, that's a quasi-species because they don't all have the same sequence. COVID reproduces in a way where it's very tolerant of mutations, as we discussed in the beginning. So even though there are sort of error checking mechanisms and so on, the mistake that the experts made about COVID evolution was they confused the mutation rate with the evolution rate. The mutation rate for the virus is relatively low, but the evolution rate is blazingly fast because it tolerates all of these different kinds of mutations, which was what we pointed out in the summer of 2020. So it's very, very accepting of mutations in its sequence. And so as a result, there's an enormous variety of viral sequences out there. And so actually the name of our blog and the name of this YouTube channel, fun fact, is Monkeys on Typewriters. And it's named for this. Think of it as a quintillion as a quintillion sized horde of monkeys on typewriters. A quintillion monkeys on typewriters typing away. And that's what they're doing all the time, trying to defeat what we throw at it. So it's a very, very large swarm. And so as a result of that, it evolves very quickly. And we've seen that as countries have dropped mitigations and so on, we've seen the rate of evolution has accelerated. This is actually from none other than our distinguished co-host today, Ryan Gregory. And what we're looking at over here is sort of the next strain sequences. And what you can see in this phylogenetic tree is how quickly COVID has been evolving, SARS-CoV-2 has been evolving. So if you look at the original strain, it's in here somewhere. Where is it? It's right here, right? Is that right? And then alpha was the first wave. Then you had all of these variants. Then you had Delta show up. And Delta showed quite a bit of diversity. When you had Omicron show up, that was a big jump in terms of sequence diversity. But then all of these have been Omicron since then. So if you now look at this where it is today, this is from December 2023. If you now look at it today, it's sort of like one more sweep around here in the last year and a half. So it's evolving blazingly fast. All of the evolution it did in the first year of the pandemic is right here. And in contrast, this was an equivalent period of time. This was 2 years over here. So the spring of 2020 or the end of 2019 to the end of 2020 was right over here. And then this is 21, sorry, 22 and 23, to give you an example. So it's evolving faster than ever before. And one of the papers that we showed was that, never mind that. So one of the papers that where we worked out some of the math on this was we showed that vaccines alone will not have an impact on the evolution of the virus. So what we looked at was, so if you had a 100% effective vaccine or a 70% effective vaccine, what level of vaccination would you need in the population to slow down the virus? And so the white parts or the paler parts in the graph are the parts where the virus has actually been slowed down because the transmission of the intermediates that were intermediate mutants has been impacted negatively because so many people, enough people have been vaccinated in the population. And if it's dark red, it means that the vaccine's having no impact. So what you notice over here right away is that you, even with a 100% effective vaccine, you really need to be vaccinating quite a lot of people to be able to make it work. And so if you have sort of, say, 2% of the population vaccinated every day, then you can end up blocking a lot of fairly transmissible intermediate variants. Now remember that SARS-CoV-2 right now has an R naught of somewhere between 5 and 9, which means every person it infects can infect 5 to 9 other people very roughly. So what this graph shows you is that the virus has a lot of headroom with the intermediate variants. Essentially, in order to be able to get the transmissibility of the virus down to a point where it's really unlikely to transmit from one person to the next, you would need to be vaccinating 2% of the population every day, roughly. And then you can block variants that have about a 4 or not. So that's basically everyone getting vaccinated every 50 days, which no one's going to do. And that's with a 100% effective vaccine. With a 70% effective vaccine, you can forget it completely. So the point is that vaccines alone were never going to be enough to slow the evolution of the virus. They were not enough to slow the evolution of the virus in 2021, as we pointed out at that time before the event. And so what this shows you is you can't rely on the vaccine alone. In the next episode, we'll talk about how you can actually fight this virus. But let's just say vaccine relax is not enough. I'm going to skip this, but this makes the point more deeply. You can go find this paper. It's an open access paper. Now, another point that's worth pointing out is that the virus doesn't always evolve to become milder. We looked at the previous two series on both on the history and on the biology of host pathogen interactions during pandemics. And what we saw was that essentially when you have an emergent pathogen, as it goes through a pandemic phase, the virulence can increase or decrease. Well, that's already been seen with SARS-CoV-2 as well. In addition to what we saw in those decks where we looked at the plague, we looked at myxomatosis in rabbits, you can also see this with SARS-CoV-2. So here what you can see is that when we went from the ancestral strain to Delta, there was a big jump in the case fatality rate. Later on, that case fatality rate declined, but the chance of long-term symptoms jumped as well. Now, what we're seeing increasingly is we're seeing lower acute infectiousness, but we are seeing longer-term symptoms. A paper that we put out there has more to say about what the long-term trajectory of evolution of infectiousness is, but we'll get to that in a little bit. The bottom line is where we are today, we're in the Red Queen's race with SARS-CoV-2. You have repeated waves of infection. You have this sort of cycle of sort of evolution and mortality. So you may look at that and say, but we're done with mass mortality, are we not? Well, the point is, until you either have extinction of the host species or elimination of the pathogen, you're still in this race. So that might sound weird, right? Like that's implausible. Like we're done with the pandemic, right? Aren't we done with the pandemic? Isn't the worst of the pandemic behind us? Well, not really. Or I would say not necessarily, right? So if you look at the definition of an endemic disease, it's a disease with wide geographic, of a pandemic disease, in fact. Pandemics have wide geographic extension. The disease moves around. You have high attack rates. You have explosive growth. You have minimal population immunity. The strain of the pathogen is novel. We saw with plague, which was endemic in the population. We saw multiple plague pandemics that lasted for centuries because the individual plague pathogens that caused those pandemics were novel, even though plague as a whole was endemic in the population. Um, and then, yeah, infectiousness and contagiousness, right? So when you look at the definition of a pandemic disease, SARS-CoV-2 ticks pretty much every one of these boxes, right? Um, and this is sort of something we all know, right? Michael Osterholm in 2009 said that a pandemic is basically a novel agent emerging with worldwide transmission. That more or less defines every new variant of SARS-CoV-2 that comes through, because when it comes through, by definition, it's defeating the pre-existing immunity. And so it comes in with a high degree of transmissibility. The severity of the disease we know is mild because it's Omicron. We'll talk about that severity in a minute. But generally speaking, every new wave is entering the population with with a low degree of population immunity, and that's why it takes off. So we'll talk about what that means in a practical sense. But bluntly put, the opera isn't over until Brunhilde sings. And so where we are today, we're very much in the thick of the pandemic because nobody is making any attempt to slow this thing down. So it's running on whatever time scale it wants to. It's evolving faster than ever before. And there are no measures in place, uh, as we've discussed, there's no impediment uh, in SARS-CoV-2's way. And this is a worldwide phenomenon. So that begs the question, well, we talked about the predictions that we made that have come to pass. And so then you may find yourself asking the question, do you have predictions about what happens in the future? And unfortunately, we do. Um, so we showed a number of years ago that there's no selection pressure favoring mildness. So over here, in this particular paper, we looked at what would happen if you had SARS-CoV-2 being endemic. What we noticed was that the transmission curve of SARS-CoV-2, this is from published data, kind of looked like this. This was in 2021. So the transmission curve is such that most of the transmitting is done by about day 10. But in 2021, with acute death, most people were dying around day 15 or day 20. This is for acute death from SARS-CoV-2. This doesn't account for the fact that now we know SARS-CoV-2 will often lead to infections that can cause death years later, months or even years later. So even just looking at acute death, the virus does most of its infecting before it does its killing. And you can see that over here, which is if you had a virus that was killing at day 50, it shows no loss of transmission intensity, and so on and so forth. If you go backwards, you notice only if the virus killed in the first few days would you see a loss of transmission, uh, effectively. So we then asked the question, if you look at the duration of natural immunity in months, right, and you looked at the infection fatality rate, what kind of yearly death rate would you see? And what we noticed was that the highest previously observed infection fatality rate, with our best guess estimate of natural immunity lasting for about 18 months, is capable of causing 800,000 deaths a year. So if we just went back, if we had a variant that showed up that just took us back to the highest previously observed IFRs, we would already have in the US close to 1 million deaths acutely within a single wave. So we haven't gotten to that point yet, but the point is that the virus has accessed those IFRs before. And the point that this paper makes is that there's no real cost to the virus accessing those IFRs. So if it's happening because of some biology, and this is what people say when they don't know, when they have no particular reason for it, but if there's some biology that prevents the virus from accessing higher IFRs, well, that'd be good to know, because if you simply look at the IFRs as a random draw from a distribution, there are some other implications, and we'll get to those implications in a bit. But what this suggests is that if we magically lost immunity to SARS-CoV-2, if a new variant came across that made us lose immunity to SARS-CoV-2, and we had a Delta-like variant, say a Delta Cron, that was both highly transmissible and also made its way to the lower lungs, as Delta did, then you would see massive waves of death. In a different paper, we showed that strongly immune evasive variants can cause both the fatality rates and transmissibility to jump. So what you're looking at over here is the size of the immune evasion shift and the R naught. And what we noticed was that strains that are strongly immune evasive will simultaneously be highly transmissible because they've lost that barrier to transmission and also have a higher IFR. Even if you assume that IFR comes back to what it was before we had a buildup of population immunity, because right now the acute death rate of COVID is very low, and that's because we have a buildup of population immunity between people getting vaccinated enough times and between people getting infected enough times. Acute death rates have gone down. The point is, even that acute death rate reduction, which has come at the cost of repeated infections mostly, is vulnerable to a big jump in the immune evasion. And what we showed over here was that such a variant, a massively immune evasive variant, would both be highly transmissible and substantially more deadly than what we're seeing right now. So you would end up with a massive wave of infections. And by the time you realize that that massive wave of infections is coming with an elevated infection fatality rate, it's too late because, as we saw earlier, most people get infected. The virus does most of its infecting in individuals before it does its killing. So you'll be well into a wave before the hospitals start filling up. And from that point onwards, there's not much you can do to change the course of events. The morgues will eventually fill up. And later on, all that will be left is for people to, clutch their pearls and say, no one could have predicted this. So this is sort of where we are right now. We've also shown in a different paper that the risk of co-circulation of multiple strains remains on the table. I won't go into this in a lot of detail, but this is a paper we published a couple of years ago, and it shows that serotypes could very easily form with SARS-CoV-2. So serotypes are really bad news because it means now you'd have to have two vaccines for SARS-CoV-2 instead of one. In other words, if you were infected with one serotype, you could be infected with the other serotype simultaneously because the one serotype would not provide immunity to the other. Now, is it possible that SARS-CoV-2 has formed serotypes already? Yes, it is possible. We don't have enough granularity in what we see right now to be able to know that. But in general, we think one of the reasons why serotypes haven't formed so far is because of the residual protection that people get against ancestral strains when they get vaccinated at some frequency. If we drop the frequency of vaccination to the point where it's all natural immunity, then yeah, your likelihood of serotypes improves if you're playing the game for the virus, which I firmly believe that there's experts out there playing on team virus. So if you're trying to get the serotypes going, continue to drop the recommendations for vaccination. Now, one of the things that we pointed out years ago was that long-term infections are an efficient source of evolutionary progress for the virus. And this next strain image, which shows you the phylogenetic trees of the virus, points out that this is kind of what's been happening. If you look at the big jumps like Omicron didn't originate from Delta, which was the viral variant that was prevalent at the time. It reached back into the ancestral tree and sort of branched off from a much older version of the virus. We now know that's very likely because Omicron was incubating in someone in South Africa with HIV who had had that infection for a matter of years before it broke into circulation. So we've seen that repeatedly. It's not just Omicron. We've seen this with other variants as well. They tend to jump back in the lineage. They don't. So, for example, BA6 over here that was succeeded by XBB. XBB didn't branch off from BA6. It came from a prior infection that was some time in the past. So this kind of long-term infection-driven evolution has some implications for us as well. So intra-host evolution is driving the bulk of viral evolution, and you can see that in this particular set of graphs. What you see is that the relative fitness of these individual strains, which are colored now in these different colors, the relative fitness of the strains has made big jumps over time. And so these kinds of jumps in terms of relative fitness were associated with big divergences in mutational sequences. So when a new variant shows up, it'll have like 50 new mutations. And so we asked the question, okay, well, how is this working exactly? Because you'd think that when a virus is evolving inside a patient, it must be operating at a disadvantage, right? Because it's being selected for on the basis of what helps it proliferate within the patient, not what helps it proliferate in the population as a whole. And so we asked that question looking at an evolutionary model. And what we found was that persistent viral evolution is likely, persistent infections are likely to drive viral evolution by increasing the genetic diversity between hosts. So over here, we simulated SARS-CoV-2 infections in an agent-based simulation. And we asked the question with longer and longer infections, what would you see in terms of the diversity of the genome sequences? And what we showed was that with longer term infections, what you're likely to end up seeing is a bigger and bigger jump in genomic distance. And this is bad news because the bigger the jump, the more genomic diversity you have. There's a result from evolution that says, from evolutionary theory, a formal result that says that the extent of genomic diversity determines the rate of evolution. Fisher's fundamental theorem. So in other words, the more diverse a population is, the more rapidly it evolves. And this is sort of intuitive in a certain sense. And what we've shown in work that we haven't published yet is that these figures, by the way, are from work that we haven't published yet. It's a collaboration with Ryan Gregory, Deborah Van Egeren, and Madison Stoddard. And what we're showing in this upcoming paper is that persistent variants are more likely to be evolutionarily successful because viral evolution favors persistence at a population level. So essentially what this paper is demonstrating is that there is a ratchet, there is a drive towards longer term infections, and there is a drive towards infections that therefore generate more diversity. Longer term infections generate diversity. And so once you get on that treadmill, you're going to keep going in that direction. And that's sort of what we've seen at this point. Also in this soon to be published paper, someday to be published paper, we've shown that intra-host and between-host fitness are just correlated enough to be able to create trouble. The correlation coefficient between individual mutations that impact intra-host fitness and between-host fitness in the viral spike protein is shown in that table on the left. And what we show is that the correlation coefficients are somewhere between 0.1 and 0.5, or 0.05 and 0.5. When you get up to 0.1 to 0.5, that's enough of a correlation that there are going to be certain mutations that benefit both the virus when it's inside the host and the virus when it's transmitting from one host to the next. So in other words, viruses that are evolving over a long period of time inside the host are going to start racking up mutations. Not all of those mutations are going to help it jump to a new host, but enough of them can help it, enough of them can pile up so that the virus is not only able to propagate efficiently inside its long-term host, but can also propagate efficiently from one host to the next. The simulations on the right kind of illustrate this idea slightly in a slightly more direct way. All we're doing is we're taking the correlation coefficient on the left and saying, how would this behave in a population level when you are looking at transmissibility? And the answer is, it actually helps the virus quite a bit. You can end up, weirdly enough, with a virus that has been incubated for a long period of time, has accumulated a ton of mutations, and is now intrinsically more transmissible, leave alone the fact that it's also going to be more transmissible because it's defeating more of the immune system. So in other words, these long-term infections are a problem. And we're sort of, the way we're dealing with long-term infections now, the strategy, the public health strategy we're using is the same as the public health strategy for everything else, which is pretend it's not happening or ignore it and it'll go away. And what this suggests is it's a ratchet. It's not going to go away. There are going to be consequences as a result of this. When you take a problem and you ignore it, the problem doesn't always go away. If you're sitting at your couch watching TV, eating your TV dinner, and you smell a burning smell in the house, and you say, never mind, I'm going to wait till I finish dinner before I deal with that burning smell. If you wait long enough, eventually you won't have to worry about that burning smell. The situation will resolve itself, but it may not resolve itself in a way that's favorable to you. For SARS-CoV-2, there are multiple evolutionary pathways to higher virulence, in fact. We've seen a lot of these things happen already. The boxes in red are changes that have happened already. These are changes that both increase virulence, acute virulence, and also increase transmissibility. And what you can see over here is that there's many of these changes that we've seen already in past viral variants. This is a preprint of ours from a few years ago. And what the point we were making here is that it's when people say there's no biology that the virus can access for increased virulence, well, actually, previous variants have shown certain biological phenomena of certain biological properties that were both beneficial to it from a transmissibility standpoint and caused higher virulence. For example, with Delta was both more transmissible than prior variants and was also quite a bit deadlier. So these things can happen. The fact that they haven't happened yet doesn't mean that they won't happen. You can walk down a street um, in a bad neighborhood for 20 evenings in a row. If you get mugged on the 21st evening, uh, saying that you hadn't got mugged the previous 20 evenings won't is doesn't really sort of um, justify what happened to you. So we're rolling the dice on this thing. And it's worth pointing out, and we had discussed this in a previous episode, but it's worth pointing out that coronaviruses have a certain amount of innate immunity. We have a certain amount of innate immunity to coronaviruses. So coronavirus fatality rates vary very widely, right? If you look at sort of the historical coronaviruses, which we talked about in an earlier episode, they've been around for millions of years. They predate the existence of humans. But newer variants, newer coronaviruses, such as SARS-CoV-2, SARS-1, as well as MERS, have relatively speaking, really high fatality rates. Now, it's true that even the common cold coronaviruses, which are the first four on this list, can be deadly in certain populations. But in general, most people will not die of the common cold, right? But the fact is that as a family, coronaviruses are not all the common cold. I'm just saying something that's very obvious here. So all of these things are not that distant from each other. If you look at the phylogenetic tree, MERS is not that distant from common cold coronaviruses relative to SARS-CoV-2. SARS-CoV-2, the distance between SARS-CoV-2 and some of these common cold coronaviruses is equivalent to the distance between certain common cold coronaviruses and each other. So again, and SARS-CoV-2, as we know, is evolving rapidly. So it's continuing to explore that space. The larger point being that coronavirus fatality rates are not locked in. Now, as I mentioned, we have a certain amount of evolved resistance to SARS-CoV-2. And we talked about why having evolved resistance is good for us for the common cold, but is not good for us as a public health strategy. There is a signature for selection of coronaviruses from an ancient pandemic. We discussed this data previously. What happened here was they looked at essentially human genes and they asked the question, where are the virus interacting proteins? And what they found was there's a number of virus interacting proteins that show strong selection pressure that are associated with human common cold coronaviruses. And what they were able to find was that these resistance markers are present only in some populations and not others. So what they showed was that, for example, South Asians don't have that. The European populations don't necessarily have that, but you see it in spades in the East Asian population. So East Asian populations are very strongly enriched for coronavirus resistance genes. So when you look at sort of how long this took, this happened about 1,000 generations ago in that population, and it lasted for about 200 generations, roughly. So if you look at sort of the width of that and over time, it lasted for, roughly 200 generations. So 200 generations is a tiny chunk of time. If you had a coronavirus sweeping through the population for the next 200 generations, in other words, the next 5,000 years, roughly, give or take, yeah, that's roughly where you would be. So it's not a great strategy for actually dealing with the public health threat to let evolution do its thing, because we've discussed before that evolution will not do its thing with the virus. It can do its thing with us, but we have to be willing to wait long enough. If we're patient enough, I suppose we could evolve resistance to SARS-CoV-2. Here's another paper that shows the evolved resistance to coronaviruses. And actually, we've already seen that there are certain haplotypes that are less likely to experience acute COVID. So again, if for those of you, out there who are the Darwinian, neo-Darwinians among us, this is great news because as long as you have that gene, then you're all set. But in general, this will take your descent. It won't be you. It won't be your descendants. It'll be, 50 generations later or 100 generations later, because the odds ratios still are not that good if you look at it. Your risk with this only goes down, 80%. So you're still at risk if you keep getting the disease, or you're not immune to it. You're not intrinsically resistant. You're simply less likely to be vulnerable. So what it comes down to is that this kind of an approach of saying that the virus doesn't have these properties or does have these properties is something that we've seen public health do the whole time, right? We wrote a blog post about this. It's called calm mongering. There's a whole name for it essentially, and what it is basically experts going on TV or going, writing articles that explain to the public that there's no reason to panic. So we've had many such messages. We were told that there was no reason to believe the virus was airborne. We had no reason to believe that the virus could kill the young and healthy. We had no reason to believe that the virus could even infect children. When it became obvious that they could infect children, then it was like, well, there's no reason to believe that it harms children. We had no reason to believe that the virus can cause reinfections. When we saw reinfections, then we were told that the reinfections were milder. When they were not milder, then we were told, that there will be no long-term health harms. There's been repeated waves of calm mongering. Every time something gets proven wrong, there's a new line of defense set up with a new lie. And we've been through this for years and years. And so when we hear people talking about it now, when you hear the experts talking about it, we always hear, oh, well, it's become mild or it's become milder or we are now immune to it. The pandemic is over because essentially long COVID frequency is decreasing. All of these things make sense if you're not paying attention to the data or you're not paying attention to the facts. So they're really designed for low information people because the data is still out there despite the best efforts of people who've been trying to hide it. And the data is fairly clear. None of these things have actually come to pass. Today, you could say there's a fresh set of concerns that you could raise and you would be accused of fear mongering, right? The possibility that the virus can cause cryptic organ damage, the possibility that it can form serotypes, that it can become more virulent over time. Each one of these things is sort of a potential risk. Now, the way the risks work is, by definition, the risks that you have that I've listed over here, some of them sound really freakish, like societal collapse or promoting cancer progression. But these are all plausible risks. And saying that they haven't come to pass yet is not very useful. It's just about as useful as it was when we were told that these risks were not worth considering unless they came to pass. We've already been through this process where a risk once identified is dismissed because it hasn't come to pass yet. Once it comes to pass, then the next thing that people will say is no one could have predicted this, which is a tagline of Ryan's, if you follow him on Twitter. Or we're told that there's no reason to worry about it because of this other factoid. And so the risks that are on the table now, because we ignored that previous set of risks, because we calm-mongered our way as a society out of that previous set of risks and did precisely nothing about the pandemic, now, as a result of 5 years of inaction, we have a fresh set of risks, and those risks are on the table now over here. We've talked about the risk of cryptic organ damage to individuals. We've talked about individual health risks, but there are also public health risks. You could end up with very dramatic outcomes in a short period of time if we roll snake eyes on evolution. And I think this is an important point to realize, is that all of these things have not come to pass. And it's also important to realize that if these things do come to pass, we will be told that they could not have been predicted. But the essence of risk management is to take risks seriously and to move to mitigate them or to move to address them, not to explain to everyone that the risk is not something that they need to worry about. There's a Nation article by Martha Lincoln and myself from a few years ago where we said that the coronavirus doesn't care about your feelings. It still doesn't care about your feelings. And so a lot of what public health has focused on is managing the emotional context of the pandemic rather than actually doing anything about it. So we're still stuck in this situation where everybody is getting infected at some frequency, the virus is evolving rapidly, and there's a whole battery of plausible risks on the table, which I've discussed here. Now, some of these risks will be discussed in this presentation going forward a little bit, but also some of these risks we've written about in the past. For example, the serotype risk, there's a paper on it. Being more virulent over time, there's a paper on it. We have a paper coming out on the risk that it may become more persistent over time. So we'll keep talking about some of these things as we go forward. So it's worth thinking this through. Like, here we are right now, and if the only strategy is to let COVID keep, doing its thing, then where does this end? At some point, are we going to, end up throwing a ticker-tape parade for the four horsemen? Like, what exactly is the strategy? What is the end game, right? So from the end game standpoint, if we continue this sort of let it rip strategy, from the end game standpoint, there are two different strategies to worry about, right? And one is the big explosive wave that happens, and we'll talk about that one more time. But before we get there, let's think a little bit about the other possibility, right? That sort of this is the way the world ends, not with a bang, but with a whimper. So an attrition-based outcome that is worth thinking about for a little. So many diseases have long-term health effects, right? HIV, chickenpox, EBV, polio. EBV is mono, but also EBV increases your risk of multiple sclerosis by I think 17 or 42 times. I forget what, but it's a very large number. An acute course of polio will lead you to believe that it's a mild disease because in 99% of the population, it's a fever that lasts a number of days, and then you're fine. In 1% of the population, you do get paralysis. In that 1% of the population, I believe about 1 in 20 dies of polio. So the long-term case fatality rate for polio is extremely low. The medium-term risk of paralysis is extremely low. 99% of people get polio and then they're just fine. It's just, it's a fever. HIV, acute HIV is a walk in the park. It's basically like the flu, maybe 3 weeks or so, 2 to 3 weeks of a flu-like symptoms, and then you're done. But again, there are long-term consequences of HIV, as we all know. So we don't fully know what the long-term consequences of SARS-CoV-2 are, but we know there are long-term consequences, and we know there are many long-term consequences. Unlike these diseases, SARS-CoV-2 is not just limited to one organ system. It hits all organ systems, and persistent infections occur in a very, very non-zero percentage of the population, as I said, between 1 in 5 and 1 in 7. Even if you drop that by one order of magnitude, this is still a far more consequential disease in terms of long-term health effects than polio is, or EBV is, or chickenpox is. We know that if you don't take precautions, you will get SARS-CoV-2 repeatedly. Some fraction of those infections will be persistent. And we know that those untreated persistent infections, at least in some cases, have been documented to be progressive, have ended in the patient's death. This is documented, it's not debatable. And individual sequelae show up at their own rate. So we don't really know at this point. For those people who've been infected already and have persistent infections, we don't know what the long-term consequences are in those people. And oh, by the way, we don't have any plan to deal with those long-term infections except to ignore them right now. So there is no plan at all when it comes to long COVID treatments, when it comes to antivirals for people with persistent infections. There's very few options available right now. I suppose there are supplements that RFK and his friends are selling. But beyond that, there's not much else out there. So what is the long-term game plan here? We know, meanwhile, the virus will not do anything to help us out in this front because persistence within the host is in the virus's evolutionary interest. The longer it persists and if it can remain infectious, it has a huge advantage in terms of propagating through the population. So we can expect over time that anything that drives transmissibility will function as a ratchet, just as SARS-CoV-2 became more transmissible dramatically in the first year or two because transmissibility was favored. Similarly, now we can expect to see that persistence is favored over the long term. And so you're going to end up with more and more people with persistent disease. At some point, that's a slowly increasing health burden of COVID. At some point, that health burden will become insurmountable. So we don't know how much additional disease burden we can actually cope with as a population. So this is something for us to keep in mind as a potential risk. Again, the way that this risk is being dealt with right now is it's simply being dismissed. But the point is the risk still exists. So we could also ask other thought questions, right? One thought exercise is, well, what if SARS-CoV-2 causes cancer? We have a series on this. It's not that theoretical. There's a whole YouTube series on this. You can make a strong case from first principles that SARS-CoV-2 causes the same cellular insults as viruses that are known to be oncogenic. So there are 7 viruses known to be oncogenic. EBV is one of them. HPV, hepatitis B, hepatitis C, HPV as well, Kaposi sarcoma virus, Merkel cell carcinoma virus. So all of these viruses are known to be carcinogenic, and they cause certain changes at a cellular level, such as double-strand breaks. Those double-strand breaks are unrepaired. They impair the cell cycle checkpoints. that deal with DNA damage repair and eventually lead to chromosomal instability. This is a common mechanism for carcinogens in general, and we know that the viruses that are known to be carcinogenic operate by these mechanisms. We know that these insults occur in these viruses. So SARS-CoV-2 also ticks all these boxes, by the way. So, you can unambiguously propose as a hypothesis that SARS-CoV-2 is carcinogenic at this point. And then people will say, well, but what about the cancer data? Well, we know the cancer rates were rising before the damn pandemic. We know they were rising in the 2010s. So you have a rising baseline. And now you're going to be able to, now you think you're going to be able to tell that SARS-CoV-2 is causing an increase in cancer rates. Well, it won't help you because the line is not flat. The line is tilted upwards. So now you have to look at the 2020 to 2025 data for cancer, which, by the way, cancer takes decades to manifest, not, it doesn't happen overnight. So you'll have to look at the post-pandemic start data and say, well, at this point, there was an inflection in that curve and cancer rates went up. So if you're looking to dismiss the possibility that SARS-CoV-2 causes cancer, I have one sentence for you. Just keep saying, where's the epidemiological data? You can play that game for 25 years in the same way that people played that game with cigarettes for a long time. And cigarettes have 100-fold increased risk of cancer. So the whole point that we should wait to see until epidemiological data says that SARS-CoV-2 is carcinogenic before we do anything is moderately, insane. Because by the time we know that SARS-CoV-2 causes cancer from the epi data, know in air quotes, everyone will have had SARS-CoV-2 infections maybe 15 times. So we have a little model that I threw a thread on, and we'll link to it in the comments to this video. Greg Kirchoff built a model. If you look at it as a bell curve of cancer incidence, what happens is that when you have a certain detectable burden of cancer, there's a bell curve of delayed cancer incidence that happens for all the previous years up to that point. So by the time you have a certain detectable burden of cancer, let's say a doubling of cancer cases causes people to panic. This is your baseline rate of cancer. We know at a baseline, 20 million people get cancer a year. So if that rate doubled to 40 million people a year in the US, what you would end up seeing is, okay, well, there'd be panic at some point, right? Well, let's say that this is the panic threshold. By the time you hit this panic threshold, all these additional people's cancers, all these additional cancers will show up in the years following the point that panic threshold was hit. So by the time you get to 80 million extra cases of cancer that happened due to SARS-CoV-2, and you say, oh yeah, this is why, you'll have another 800 million. I'm not saying that this is the burden of cancer that we can expect from SARS-CoV-2. That's not my point at all here. My point is, even if you had a certain attributable burden of cancer that was visible for SARS-CoV-2 at a certain point, and that would only be visible if the baseline was flat, which it is not. It is rising, and it was rising before the pandemic. And without noise, this baseline is not without noise. The real baseline is noisy. So even in this best case scenario, if you could spot a certain burden of cancer and say, oh shit, SARS-CoV-2 is causing cancer, and even if on that day, you shut down all future SARS-CoV-2 transmission, so no one ever got a COVID infection after December 31st, 2038, you'd still have 10 times as many cancers following. So you can play with this. It's a tool. It's a simple tool to show you that when people argue for the delaying tactics of waiting for epidemiological data, they are simply building in a locked-in burden of disease that will last for decades afterwards. We've done this for cancer, but this is just a thought exercise right now. Cancer as a risk for SARS-CoV-2 is plausible, but there are dozens of other plausible long-term health hazards for SARS-CoV-2. You could do this same graph for 10 other different organ systems. It's not just a question of cancer, it's a question of brain damage. It's a question of damage to the circulatory system. It's a question of increased risk of liver failure and kidney failure. There's all kinds of things. But all of these are delayed risks. And all of these delayed risks, by the time we see the signal in the epidemiological data, it'll be too late to do anything about it because we'll have a much larger burden of disease for decades to come afterwards, very likely. There are very plausible scenarios where this attrition-based outcome of let's let people get sick with SARS-CoV-2 all the time not only has a rising burden of disease because each individual gets infected more and more often, and therefore each time picks up a delta increase in the risk of persistence, but also has a rising burden of disease for a second reason, which is that the virus is evolving to become more persistent, and for a third reason, because the burden of delayed disease from any one of these organ systems being damaged could take years to manifest. So whatever burden of disease we're seeing today, of long-term disease we're seeing today, and it's not small, all the factors point to that being larger over time. We just don't know how much larger. And we don't know how much larger a burden we could actually absorb as a society. But, here we are ignoring it. And I think it begs the question, is this the way you manage emergent risks? Is this a smart idea? Like, how many other emergent risks do we manage like this? How many other emergent risks do we have do we have a clue that this virus may be causing? And in general, when you manage risks, whatever risk it is, whether it's a hurricane or whether it's a risk of a fire, la 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 is only lasts as a strategy for so long. And so a good question to be asking at this point of our public health authorities is this a smart way to be managing a risk? Um, and this is only one half of what could go wrong with SARS-2, right? There's also the other half, which we've alluded to, which is the so-called black swan event, the risk of sudden catastrophe. So we've talked about two ways to access increased IFRs. One is through immune evasion. We talked about how immune evasion could lead to a strain that is at once more transmissible and more deadly, and also altered innate virulence. So we've talked about the pathways by which SARS-CoV-2 could access higher virulence. The fact that it hasn't for a while doesn't necessarily mean it can't, because it's done that before. And we've also talked about the way in which punctuated equilibrium with long-term infections is a very efficient way of generating new variants that have large jumps, both in the mutational or sequence space and also in immune evasion. So we also know that SARS-CoV-2 has had altered host tissue tropism. For example, with Delta, SARS-CoV-2 had increased tissue tropism for the lower lungs. So the Deltacron scenario remains on the table, which is a highly immune evasive, highly transmissible, but also lower lung colonizing variant. If something like that came along, we talked about it already with a 3% fatality rate, something like that in the US would minimally lead to, 7-figure mortality. Again, the strategy for that is to ignore it, right? These would be the global numbers for an IFR of 1 or 2%, which the virus has seen before, or 10%. Now, put it in context, 10% seems high, but 10% is what SARS-1 had. MERS is 34%. So in scale, these things would begin to approach the 1918 pandemic or exceed it trivially, even in terms of percentages. And so the main thing with a highly transmissible wave is that a wave like that would come through in weeks. Most people would fall sick before we knew what the IFR was. So again, the only thing left for people to do would be to rub their hands and sort of let us know that this was not predictable, that things will be fine now because it's not going to happen again. But again, if the strategy is to ignore it, the question then becomes, this is a risk. What could happen as a result of ignoring it? So there's a name for this. It's called non-ergodic gambling. So when people gamble, the house always wins, right? The house always wins, weirdly enough, not just because the payouts favor the house, but because eventually the gambler's source of funds runs dry. For everyone who gambles except potentially Elon Musk, if you stay at the casino long enough, you will bankrupt yourself because eventually you run out of cash. It's true that you could get a win later on, but you'll never see that win because you need to leave because you ran out of cash. So if you think of each variant wave coming with its own IFR drawn from an underlying distribution, the distribution of IFRs is a really key question. What does that distribution look like? We've had 10, 20 waves so far. Is that enough to estimate a distribution? We'll dig into that question in a minute. But we know that future variants don't have to be milder. Now, this sort of non-ergodic gambling, where you eventually go bankrupt because you run out of money, is an analogy for what you can expect to see with SARS-CoV-2 in the sense that if you have enough waves coming through, at some point you're going to hit a really bad wave in theory. So the question is, how likely are you to see collapse like this, right? Now, the more times you draw from a distribution, the higher the max IFR will be. That's just a statistical property of something called the extreme value distribution. In other words, the more people walk through a room, the higher the height of the tallest person who walked through the room was. Common sense, right? It'll never go down. The tallest one will always, the height of the tallest person will always go up. So on this disease management trajectory, human extinction is a plausible risk. Now, that sounds very extreme, right? It's a, I'm saying it's a black swan event, right? So the black swan event concept comes from Nassim Nicholas Taleb, who wrote this New York Times bestseller many years ago, a number of years ago, called The Black Swan, where he talks about these risks that are sort of not visible to people necessarily, but can be catastrophic. And so Taleb talks about black swan risks in the context of financial markets and so on. So sure, you can say, well, it's kind of a bit melodramatic to use Taleb's idea of a black swan, applying it to a pandemic context. So actually, Taleb's the one who has done that. So in 2020, at the beginning of the pandemic, he pointed out that over time, in a paper that he published, over time, the risk of a black swan event goes up, right? So essentially what he said was, this is the abstract of the paper. And what he says is, while there's a very high probability for humanity surviving a single such event, a single pandemic event, repeated risks over time are going to be, are will increase the risk of a black swan event, essentially. Essentially, the sentence is, while repeated risks can be taken by individuals with a limited life expectancy, ruin exposures must never be taken at the systemic and collective level. And so this is the whole point, that this is where risk management needs a more proactive forward position. You shouldn't be looking at what's the likelihood of X or Y risk. You should take a proactive forward position, because traditional statistical averages are invalid in the case of black swan or non-ergodic scenarios. Right? In the casino, the point is don't gamble, right? And for pandemics, the point is exactly the same. So this is a fairly detailed paper, but what Taleb and his co-authors did in a different paper was they looked at the death tolls from different diseases, from different pandemics over time. They looked at all the historical pandemics they could find. And what they were able to find was, when they looked at this and they built the statistical distribution, and then they analyzed it, looking at the first, the second, the third moment, which is essentially the first moment is a mean, then the second moment is a standard deviation, third moment is a skewness, and so on. I won't get into it. But the point is, there is a parameter in the fourth moment, kurtosis, which is essentially how fat that tail is. And what Taleb was able to show was that you don't really see, the distribution drops off weirdly. You don't see the events that you would expect to see that had very high numbers of casualties. He measured the casualties to the same baseline. So the baseline is today's population. So the 1.4 billion or 140 million is baseline to today's population. And what they noticed was that you see this drop off. This should have been on a straight line, but it isn't. And so one of the things that he pointed out in this paper was that most likely that's because the really destructive pandemics are not recorded in history, as one would expect. First of all, they happen very infrequently, and where they've happened, we've not had access to enough survivors to maintain that record. We know that destructive pandemics have happened in history because our genetic record shows that. We know that destructive pandemics have happened in hominid history because one of the major predators of hominids is infectious disease, as we know, especially in the modern era. And we know that other hominids went extinct. So at least in some part, as we discussed in earlier episodes, there's a plausible case that can be made that infectious disease was responsible for some of those extinctions. So we know that it's, in theory, an extinction level risk. We know that the tails are fat enough that all of that may not be visible to us at any given time. Right? So don't take my word for it. This is Taleb's, an interview from Taleb in the summer of 2020. And what he says over here is, things like pandemics are really fat-tailed events, and you really should be prepared for the worst case. And the key point here is not that we're all going to die. The key point is that when you have pandemics, and when pandemics bring certain risks with them, just about the worst thing you can do with those risks is to ignore the worst case scenario. I'm not saying the worst case scenario will happen, but I'm pointing out that the strategy that we're using of ignoring the worst case risk is extraordinarily stupid, because it actually doesn't help you in the least. And if you wait long enough on a strategy of ignoring the worst case scenario, you're going to be faced with that worst case scenario eventually. Right? So the point over here that Taleb makes is that, a lot of what we've been talking about now, which is that you can very successfully make claims about the maxima. So what he talks about is the maxima in terms of death rates, but in terms of deaths, raw deaths, but we can also look at it in terms of IFRs. And so the point is that the IFR distributions are likely fat tail distributions. How does he know that? Because of the statistical analysis he did on historical data sets. And what he showed was that those IFR distributions are in fact, the fatality distributions are in fact fat tailed. So, essentially, this is not about crying wolf, according to Taleb. His point is that pandemics are fat tailed phenomena. So you can say, okay, well, pandemics are fat tailed phenomena. That's all great, but we would have seen it by now if it was fat tailed, would we not have? So Greg Kirchoff and I worked on a little simulation. Again, this is accessible. We'll share a link in the comments. Over here, you're looking at two distributions. This is getting a little bit technical in statistics, but this first distribution is sort of a standard narrow tail distribution. The one in green is one with a much fatter tail. So over here, you can slide around the median infection fatality rate. You can slide around how fat that tail becomes. The sigma slider affects the regular distribution, and the alt sigma is the one in green, the fat tail distribution. So the fat tail distribution has a significantly higher sigma, which is a measure of the width of the tail. Now, what you notice with this is if you run simulations from this in terms of populations, just drawing from this and having just a standard population doubling model, you have a certain number of waves coming through once a year. I think it's 1 or 2 waves a year. What happens is that if you have COVID, if COVID follows this purple distribution, what you'd see is that COVID doesn't really impact human populations that much at a certain level of growth. However, if the distribution of IFRs for COVID looks more like the green distribution over here, then over time population growth kind of looks like this. In other words, this is already bad news because it means it'll sort of drive population to a stasis. And if you look at it over here, you're looking at about 50% fewer human beings alive in 100 years. So this is, if you look at it that way, this is, tens of billions of deaths due to COVID. Right, so you can say, well, obviously you're going to be able to tell the difference between those two. It's readily visible, right? This is what happens when you draw 10 points at random from the purple distribution versus when you draw 10 points at random from the green distribution. Does it look different to you? This is the t-test for p-value, 0.7. And this is a second test we did, looking at not at the mean, not at the difference between the means in this case, or strictly speaking, the first test was difference between the medians. And the second test over here is a test of the standard deviation. So the question we asked first was, will the medians be different? And the answer is no. Then we said, well, will the second one be more variable? And the answer is no. So this is just one draw. If you go to the website, you can see his page shows multiple draws. So he does multiple draws of n equals 10. And pretty much almost never can you see the difference with n equals 10. You can say, okay, well, we've had 10 waves now, but surely soon enough it'll become visible, right? So this is what happens with n equals 20. Here's a typical example of 20 draws. Now, again, his page allows you to select how many draws, what the sample size is. So yes, once you get to a sample size large enough, you can indeed see that these are different. What you see over here again is that with n equals 20, the median IFR is not different. And once 20 waves are through, you'll begin to see maybe that there is a slight difference in the variability. But even here, that's not statistically significant. So even with a higher, so the point here is that you have two distributions that have dramatically different outcomes in in a 20, 50, 100-year time frame, but they don't look different in terms of IFRs. So looking at the IFRs that we've had so far with the limited number of waves we've had come through, you can't tell that much about where this is headed. Even if you had a dramatically different IFR, such as the one in green over here, which on the right-hand side is sufficient to drive humanity to extinction in 100 years, by the way. This is what it looks like with 20 samples. Your first 20 waves, yes, it's true, it looks a little more variable, but the medians look the same. In fact, in anything, if you look over here, this is a probability density function, right? So if you look over here, the one in green has lower IFRs than the one in purple. So you cannot point to the fact that a few waves have had low IFRs to deduce that the tail of this distribution must be narrow. This was Taleb's whole point, and Greg Kirchoff's simulation shows it clearly. So we are bullshitting ourselves with potentially disastrous consequences if we extrapolate from the 10 or 20 waves we've seen so far to say that just because we haven't been driven extinct just yet, nothing catastrophic is going to happen to IFRs. There's no reason to assume that. We are gambling with our futures. In fact, we're in the casino in a non-ergodic game, and the house always wins. So the strategy of letting the virus rolling the dice each time with viral evolution, letting the virus do its worst, is an extraordinarily boneheaded strategy. Not only because of the risks we talked about first in terms of attrition, but also because we could end up with a nasty surprise that no one could have predicted once again, because we don't know what the shape of the IFR distribution is, and we won't know until it's too late. Again, play with this. It's available on the website, on Greg's website, and you can play with it back and forth. You can slide these things around. What you can find over and over again is you can generate catastrophic outcomes in that graph on the right over here, with differences in IFRs that are absolutely minuscule at an n equals 10, n equals 20. Even if you look at the first 10 years, things don't look that different, really. So it's all fun and games until the shit hits the fan, is what it comes down to. So to be fair, Homeland Security professionals are aware of this. And this is an actual journal of the NPS Center for Home Defense and Security, which is actually affiliated with the US government. I'm sure we go looking for this, it's probably down already. But the point is that global pandemics are extinction level events, and people who are professionals in this field are aware of this. And so this is something that you may say, okay, who are these doom and gloom folks? This is the Naval Postgraduate Center for Homeland Defense and Security. And so I think, again, keeping in mind that the US government is well aware of this, that the US Navy is aware of this, the concept that these pandemics are extinction level events and that they're not to be taken lightly. I think we should juxtapose that with the fact that we are currently not only in a pandemic, but each individual wave of COVID can be considered a fresh pandemic. We are just rolling the dice in a fresh pandemic each time, because every time it comes through, if it's coming through with sufficient transmissibility and sufficient evasion of the immune system in order to be able to get off the ground, you're dealing with these highly unpredictable events. So how does the Red Queen's race end, right? We've talked about this graph for COVID, and we've said that, look, we're in this sort of middle loop here. And when we talked about it first, I think you may have looked at that mass mortality and said, well, that's kind of weird. But I think we've justified in this talk why that mass mortality box exists and is still a risk for COVID in a variety of different ways. So it's going to end in one of these two ways. And the question that I think is fair to ask is, which of these extinctions are we talking about? Which of these outcomes is more likely? So this is a very reassuring article from The Atlantic from a while back. The Atlantic is very good at publishing very reassuring articles about COVID, no matter what happens. That's kind of like part of why they exist. You should go back and look at who owns The Atlantic if you are wondering why The Atlantic is so pro-COVID. It's a good question to ask. But anyway, so this expert was asking the question, why hasn't disease wiped us out yet? And apparently this expert has not heard of survivor bias, because if disease had in fact wiped out the human race, the expert in question, presumably being a human being, would not have been able to write this article, right? And so survivor bias is illustrated by this graph, which many people are familiar with, which shows the bullet holes in planes that came back in, I think, B-25 Liberator bombers that came back from Germany. And so the point that people made when they saw this graph was that we should armor these portions of the plane, the ones where the bullet holes exist, because those are the ones that are most vulnerable to being hit. And actually, that's wrong, right? Because the planes that didn't come back were the ones that had the holes in these other parts. So the vulnerable parts of the plane are the ones that the planes returning don't have holes in. And so the reason why disease hasn't wiped out the human race yet is because if it had wiped out the human race at this point, we wouldn't be having these conversations. So simply using this logic that we shouldn't worry about it because it's nothing that's happened to us before is just survivor bias. There's if you have unrestricted evolution, you could think of it this way as well, that either you're going to see the virus attenuate or go away. And there's no precedent through biology or through human history for viruses just vanishing because they felt like it, right? Yes, 1918 flu went extinct, but it went extinct because people took precautions. And other clades stuck around, and those clades, although they branched off at the same time as 1918 did, as the 1918 influenza did, they were milder. The virus didn't attenuate. Precedent exists for host species extinction, for other hominid species, for other species in general. So the Red Queen's race is going to end in one of these two ways, and only one of them has natural precedent. And the question really that we should be asking ourselves is which one is more likely? When you look at other species that we examined, spontaneous viral evolution or evolution of resistance, which one happened? What you can see is basically extinction happens over and over again in other species as well as humans. So if you look at past historical pandemics and you look at viruses as well that infect other species, what you see is the pathogen has gone extinct in certain cases, but that was because of human intervention, either due to eradication or due to altered environments, or in fact with 1918 because people took precautions. When it comes to the evolution of resistance or the evolution of host resistance, that does happen if you wait long enough. But the one thing you can count on is that the virus will not magically evolve to become more attenuated. So the entire strategy of sort of running with it and saying, well, let's just see what happens, learning to live with the virus, sort of presupposes that there's a certain degree of domesticability, right? And what you can see in these examples, especially in other species, is that the impact of this is felt more on the host population than it's felt on the pathogen. The pathogen is less likely to evolve to become less virulent. The host is likely to evolve resistant. So that's a long-term strategy and not one that if you are planning on long-term evolution of host resistance, you probably don't need public health. Domesticating virus is a silly strategy. Virus evolution is unpredictable in the short term, and many pathways favor increased virulence. Attritional collapse is a possibility, as we've discussed with SARS-CoV-2. Catastrophic collapse is also a possibility. So if you gamble long enough, you're going to find out, right? You're on that FAFO curve, and the longer you keep FOing, the longer you keep FAing, the more likely you are to FO. And so that price tag for the evolution of human resistance, which supposedly is a game we're playing, is not an affordable one, both at a societal level and at an individual level. Evolution is not our friend, and viral evolution in particular will not lead to good outcomes for us. So that was all depressing and all. Let's talk about how we can still win against this thing in the next episode, because there are still ways for this to work out for us. And we've talked about how ignoring it will not get us to that outcome. But if we chose to do things about it, there are practical things that could be done at a societal level, and also at an individual level, to reduce our risk and to potentially win the fight against SARS-CoV-2.
Very interesting. I hope that most of the viewers will have seen the sort of themes develop all the way through this series so far. And one of the things that that will come up in the next part that has been seen in past plagues and pandemics is that action by human populations, by public health efforts to actually mitigate can be successful, can lead to a positive outcome, and just living with things generally does not. So I think it's very important. It's not just doom and gloom. Has been a call to action and to learning from the past, but also understanding how things move along in the future. And one of the things that happens every time a significant new variant shows up, very often, almost exclusively at this point, as a result of something that happened within a host, either a long-term infection and or a recombination event that occurred within a host. The immediate response is it's not more severe. It's still Omicron. We don't need to change our course. And we've been through this many times now at this point, right? This is now, 2025, and we've been through this over and over again. And almost, every single time this happens, the folks who track the variants and point out the importance of recognizing a new one will say, will be pretty clear, I think. We're not saying this particular thing is going to cause, a Delta wave or a first Omicron wave. In fact, that's very unlikely at this point for a number of different reasons. The issue is this is illustrating that these processes are still ongoing, and it's now established a new evolutionary lineage. And so it's about the potential, both in the short term and for that to now be something that infects someone and becomes a component of a new lineage down the road. It's just reinforcing the evolutionary potential.
Yeah.
That is just absolutely not the way these things are being evaluated every single time. It's always about, is it more severe? Is it going to cause an acute wave? And that's just not the point, right? Like, it's just not the point. So hopefully, and the time scale is one of the really important point that I see coming from the past description, the description of past plagues and pandemics, is the time scale. Like, we're 5 years in, and the idea that we know where we're at and we know what can and can't happen, despite having been wrong about all, not your group, but various public health officials having been wrong all the way up to this point, but now are very confident that it's over, when that's not even historically, been the case. And it's certainly not been the case with a number of viruses that have turned out to have long-term consequences. Some of those don't show up for years and years. Like, we've got more evidence now, I would say, in the first 5 years than perhaps we did with HIV or other things in that first 5 years of emergence. We know how severe those long-term consequences can be. So just people are messed up on the time scale and on what you should be looking for each time something like this happens. I take each emergence of a highly divergent new variant, especially if it's a result of long-term persistent infection and within host evolution, as an indicator that these processes are still happening, and that's problematic in the big picture.
In and of itself, yeah.
In and of itself. And I'm not worried about, generally speaking, the current one, NB181 is going to cause a Delta wave. No one says that, right? That's not the issue. So
yeah, I think a point that I've made before and we've talked about before is most drunk drivers will drive drunk 200 times before they wrap themselves around a tree. So every new wave is like one more drunk drive. And the fact that we haven't yet wrapped ourselves around a tree again doesn't mean that it's a good idea to keep driving drunk. And so I think the entire sort of narrative becomes, oh, here's a new variant. Oh, it's nothing to worry about. Oh, it's less severe or whatever. It's mild. It's Omicron. And then when the wave passes, everyone's like, oh, see, there was nothing to worry about there, which is all well and good until the shit hits the fan, right? It's all fun and games until the shit hits the fan. Yeah.
Yeah, and that raises the one other important point about narrative and the framing of things, because the notion that everything is still Omicron from a biological standpoint is utterly absurd, right? Like, it makes no sense. It's very much at this point. I think I tweeted recently. I said, it's like if you said, somebody's bitten by something and you said, oh, don't worry, it was just an insect. Well, there's an awful lot of insects, right? So that level of resolution is meaningless at this point.
It has no utility.
Those names haven't been, they haven't assigned any new names since basically November 2021. And even under the criteria that have been made more strict, at least two of them should have been given new names because they required updated vaccines, which is one of the criteria that would qualify. But it's just not again, it doesn't matter in some ways whether you go with pi or rho or whatever else under those criteria. The point is that that system and those official nicknames vastly underestimated the degree of evolutionary change that was going to happen, the diversity that was going to occur. And it's still being used as a way to limit an understanding of that. That's what I find really problematic. It's a derivative of Omicron. At this point, it's almost like saying it's a derivative of SARS-CoV-2, so don't worry about it, right? Like, it's not a meaningful thing at all. So we just keep missing the point. Is, I guess, the issue, right? And maybe deliberately is.
Yeah, I think that is the point. That is the point. I think what we've seen is a lot of counter programming to reality, right? That is, in that calm-mongering article, this is the point, right? That
Mhm.
essentially calm-mongering is the counter programming to reality. If reality is going badly, you bring in experts who maintain an aggressively incurious, aggressively non-detail oriented sort of view of the situation. If you had looked at the facts in the summer of 2021 and you were speaking from the facts,
Mhm.
no one who was doing that was going to say the things that the experts said then. It's not that the science changed, it's that the experts at that time should have known better and either through negligence or through outright lying did not say what they needed to say. The facts were visible in the public domain at that time. So at every step along the way, the facts have been visible in the public domain. We posted that preprint about the rebound, the variant-driven rebound, weeks before, months before Walensky was on TV talking about how Delta was such a curveball. These
Mhm.
things were in the public domain. And I think that the whole calm-mongering strategy has been to maintain an aggressively intellectually incurious, aggressively ignorant, sort of doggedly ignorant um, perception of the science, and then say, "Oh, but that's what the science said. The science changed." No, the science didn't change. You ignored the science, and then the facts became too hard to hide. That's all.
It's interesting to kind of contemplate what would it mean for it not to be an Omicron lineage as an example.
Yeah, exactly.
And it would mean reaching back, as you said, it would mean having some ancestor of a variant that was pre-Omicron. And so now you're talking essentially 4 years. Somebody has to have been infected for 4 years. Well, we had one, the most recently dominant lineage was a BA2 direct derivative, which was at least 2 years
Yeah,
of an infection. And so somehow, that's not at the threshold where you need to see it as sufficiently distinct.
yeah,
I guess they're waiting until there's a new Delta derivative before they would even consider changing that narrative, which is again absurd, right? Like it's just an absurd approach to everything. But it fits very much into this whole narrative of we're past the worst of it. Things can only ratchet in a positive direction, and I think I hope that this
yes, for the
series
virus.
and lessons from the past and the sort of actual context, the factual context of how evolution works in these viruses, especially under the conditions we're giving it, will make it clear you just cannot operate like that and hope and assume that things will be fine because there's just so many ways it could not be fine. So in any case, so that's ending a little bit on a critical and somewhat gloomy view of our current approach, but as as is very helpful, we'd like to have some suggested solutions so that we don't have that be where things end up, and that'll be what we cover in part 4 of the series. So do check out the previous uh, entries in this series, and also you heard about SARS-CoV-2 as a potential carcinogen. There's a series of videos on how cancer is triggered and progresses, and the role that viruses like SARS-CoV-2 might play. So there'll be links to those in the description also. And as always, we'll look forward to seeing you next time.
Right.
Cheers.